Today, we're diving into something really groundbreaking in cancer research called the Mozart study. We're going to break down how scientists are quite literally learning to read the genetic code of cancer to find its weaknesses. Let's just start with a really tough but crucial question. For patients who are dealing with advanced cancer, what happens when the standard playbook, things like chemotherapy, just isn't working anymore? Where do you turn then? Well, this is where a really exciting idea comes into the picture, the promise of precision medicine. The big question it asks is, what if the secret to killing a tumor wasn't a one-size-fits-all sledgehammer, but a key hidden inside the tumor's very own DNA? And that idea has a name, precision oncology. The core concept is actually pretty straightforward. It's about tailoring a treatment to the specific genetic mutations of a single person's tumor. So instead of treating breast cancer, you're treating your breast cancer based on its unique genetic fingerprint. And that brings us right back to the Mozart study. You can think of it as this incredibly ambitious project to draw a detailed map of these genetic weaknesses across a whole bunch of different advanced cancers. It's like creating a genetic atlas for tumors that have kind of run out of other options. So how'd they actually do it? Well, the process was super methodical. First, they brought in 304 adult patients, all with advanced cancers that had already been through a lot of treatments. Then, they take a small tissue sample, a biopsy, from a tumor. The really high-tech part was next. They sequenced the tumor's DNA, looking at 505 different genes. And finally, they sifted through all that data to find and classify any genetic quirks or mutations. Okay, so after all that work, getting all those patients, sequencing all those tumors, what did they actually find? What does this genetic map of advanced cancer really look like? Now, here's the first big headline number, and it's a jaw-dropper. A whopping 78% of the tumors they looked at had at least one potentially actionable genetic alteration. Now, on the surface, that sounds amazing, right? It suggests that for nearly 8 out of 10 patients, there could be a genetic target for a specific drug. And if we dig a little deeper, the study pinpointed the genes that were most often mutated. Right at the top of the list is TP53, a famous tumor suppressor, showing up in over 20% of the cases. After that, you have other major players like BRCA2, PIKCA, and KRAS. It's almost like a most wanted list of genetic targets in these cancers. But, this is a really big but, this is where the story gets a lot more nuanced. Because it turns out, not all of these genetic clues are created equal. Just finding a mutation doesn't mean you've found a silver bullet. The real trick is figuring out which ones are truly actionable. So to sort through this genetic mess, researchers use this really smart tool called the ESCAT scale. It's basically a ranking system that grades mutations based on how much solid clinical proof there is that a specific treatment works for that specific mutation. It separates the sure bets from the long shots. And when the Mozart study used this scale, wow, the picture became much clearer and a bit more sobering. Look at this. Only about 19% of the mutations were tier 1. That's the top tier, meaning there's proven value. Another 16% were tier 2 with pretty good evidence. But look at that massive third bar. Nearly 2/3 of the mutations were in lower tiers, meaning any link to a treatment is still just theoretical. So this gets us to a super important point, the huge difference between finding a potential target and actually giving a patient a treatment based on it. Let's follow the numbers down this funnel. We started up here, right, with that big, hopeful number, 78% of tumors having any kind of potential target. That's the top of our funnel. But as soon as we apply filter for only the highly actionable targets, the ones with strong evidence, those ESCAT tiers I and II, the number gets cut by more than half. All of a sudden, we're down to 35.5%. And then you have the final step, the real world. When you factor in everything else, a patient's overall health, if the drug is even available, only 20.4% of patients ended up getting a therapy that was actually matched to their tumor's genetics. We went from 78% all the way down to about 20%. That really shows you the gap between potential and practice. And there's another twist. The usefulness of all this testing is completely different depending on the cancer type. You see, these highly actionable targets were found in a good chunk of cases for cancers like breast, melanoma, prostate, and lung cancer. But for many other types of cancer, the rate was way, way lower. Now, to really drive home why all this context matters so much, we have to look at this incredible case study from the report. It's basically a tale of two tumors. Okay, just look at this. It's fascinating.
You've got two different patients, both with advanced cancer, and here's the kicker. They both have the exact same genetic mutation, a CDKN2B deletion. Patient 1 had a parotid tumor, they got a drug called abemaciclib, and the result was just, wow. They were progression-free for over a year and a half, and their quality of life improved. Now look at patient 2. Same mutation, same drug, but their tumor was an endometrial cancer. The outcome? The drug only worked for 2.5 months. Same gene, same drug, completely different results.
The study's authors really hit the nail on the head with their conclusion. They said, interpreting genomic alterations in a tumor-type-specific manner is critical. What they mean is the context, where the cancer came from, matters just as much, maybe even more, than the specific mutation you find inside it. So all of this brings us to the future. If the data is this complicated and this nuanced, how do we possibly turn it into clear decisions that are actually going to help patients? Because what the Mozart study really shows is that the key isn't just about generating more data. It's about achieving a wiser interpretation of the data we've already got. And this is where the Molecular Tumor Board, or MTB, becomes so important. This is the practical solution. It's a team of experts, oncologists, geneticists, pathologists, you name it, all coming together to talk through these complex cases. They translate the genomic reports, they debate the best path forward for each unique patient, and they help find the right treatments or clinical trials. It's about taking high-tech data and adding high-touch human wisdom. Which really leaves us with this final big question. Is the next great frontier in cancer care not about bigger data or faster computers, but simply about better, more thoughtful human collaboration? It seems the future of reading cancer's genetic code isn't just about the code itself, but about the brilliant team of people who can translate it.